Okay, next up we have Xiao Liu coming from BGI Shenzhen, who will be telling us about the Pan-Immunome Initiative.
Thank you. Thank you for a very nice introduction. So just want to check it work. Okay. So special thanks for organizer for inviting me here. So our group started to doing TCR and PCR repertoire analysis about 6 or 7 years ago. So we are started from the very early stage, including the strip9 pipelines, and including both the experimental pipelines and bioinformatic pipelines. We are optimize the pipelines, and we are set everything down, and we started to do applications, and we generate a lot of data. So we move forward to the application of this repertoire data. So today I'm not going to. Actually, we have done a lot of different applications, different disease settings and diagnosis, prognosis. But today, I won't talk about everything we did, but I want to focus on the initiative that we started, and also some of the early results about applications from this initiative. So, in order, so today my content, including the some of the very brief introduction about the pipelines, the tools, and the database we generated, and also want to talk about a very brief introduction about Pan-Immunome Initiative we are started, and the applications. So, in order to accomplish the goal of the Pan-Immunome Initiative, so we did a lot of the optimization and the development for the pipelines. So, because the Pan-Immunome Initiative relying on large batch of samples. So I think for DNA, for TCR repertoire, we prefer to do the DNA, genomic DNA. So for DNA, I think the most straightforward is the amplification, the amplification enrichment from the repertoire. So we spend a lot of time to optimize the primer set, including to using the synthetic templates to optimize the primers and to determine the best concentration of the primer set and to eliminate the bias. So we did a lot, and we don't do much for the 5' arrays, but this is just for comparison for the development of the multiplex PCR amplification. So we can see the replication is very good, and besides the experimental pipelines, we also developed an analytical pipeline which is called iMonitor. So this one's been published in 2015, and this already becomes one of the most widely used softwares in the TCR and BCR repertoire analysis. So besides these pipelines, we also, we are among the first to realize the importance of the Gemini inference. And we did make some, developments to starting from the repertoire, a rearranged repertoire, to infer the Gemini from both the B cell and T cells. So we published a paper in 2016. That's among the earliest. So the software is called AIRRR4. It's can derive or predict the TCR, BCR Gemini sequences from a deep rearranged data set. And after this publication, we'll also use the macaque, B cell receptor immunoglobulin sequencing data to infer the macaque BCR Gemini sequences. And after the prediction, we compare our inferred Gemini alleles to the previous published ones and also IMGT, and we discover a lot of the known alleles and also some of the novel alleles and genes. And then we do a phylogenetic tree, and we properly to assign nomenclature of all these alleles and genes. And we recently, this work had been done almost 2 years ago, but it took us a long time to prepare the manuscript, so it's recently published. So in order to facilitate data sharing, because we generate a lot of data, so we think that it's very important to share this data and to find a very, very good repository to store our data. So in order to share the data, we established the Pan-Immunome repertoire database, which is called PERD. So this one currently constructed. So including the users can upload their data to the PERD, and they can query the data to the PERD database. and compare the data to our data, and also to from the processed rearranged data, they can do the comparison. They can find approximately find the appropriate way to visualize the data, to visualize the comparison. And after visualization, they can draw, they can plot figures, they can download figures, and then can so. And PERD is not just a database; it's including the data processing, visualization, and comparison. So in PERD, we also currently PERD contains approximately more than 2,000 different samples. Most of samples, with the publications we have published, but there are more samples to be loaded in the PERD. And PERD, we also want to collaborate with the AIRR-seq, since they are for API, we integrate. So in PERD, not only there are some of the generated repertoire data, but also we are integrate TBA DB, which is manually curated the TCR, BCR receptor sequences. So we spend a lot of time to develop this. And so currently, this database contains more than 50,000 different records or sequences, including both the TCL receptors and BCL receptors. So regarding the different disease type or antigen type, so mostly we can see that both the infection disease, autoimmune disease, and some other. But regarding the records itself, we can say most of the records are from the infection agent, from the antigen, infectious antigen. Very few of them are from the autoimmune disease, and quite a few from the tumor or cancer type. So we did a description of this TBA-DB, and also compare TBA-DB to other manually curated databases, including BGI-DB and something else. So we can find some of the overlap, but we also curated some more records. So next, I want to see. So these are all the applications from the TCL and BCL repertoire data. So of course, the most straightforward and translation is the minimal residual disease. But we also very interested in therapeutic targets or biomarkers about cancer immunotherapy. But most, what we are mostly interested is the diagnosis, whether the TCL, BCL receptor sequences can serve as potential biomarkers for disease diagnosis, prognosis monitoring, classification, or even the early detection of disease. So this is the Pan-Immunome Initiative, the reason we want to establish this. So what is the Pan-Immunome Initiative? So major purpose, as I just said, we want to explore or investigate the potentiality of the TCR, BCR to serve as biomarkers for the molecular classification of disease, better treatment, and also for the prognosis monitoring. But most importantly, ultimately, we want to work with the non-institutional or prospective biobanks to explore the potential for them to serve as the early detection, early screening biomarkers for disease. This is not for a single disease, but for multiple disease. It's it should have a very bright future. So, to accomplish this goal, we are started to connect samples and working with the other partners. So currently we're in a stage that's something, we're in a stage of this. So in the first stage, we want to collect as more as the different advanced disease samples. For these samples, we want to do the disease diagnosis, multiple diagnosis, and also to look at the whether any heterogeneity exists and potentially subtyping or molecular subtyping of the diseases, and also to correlate the biomarker with the disease phenotypes. But in the next stage, we also want to connect more samples of the early stages. So this will facilitate us to look at any early signals about the TCR or BCR signature. So in the future, eventually, we want to collaborate with the prognostic, biobanks or cohorts to see whether it can do early detection of the disease. So it still takes some time to do so. So the collaboration model of the Pan-Immunome Initiative is we established a network which includes the hospitals. Of course, the BGI serve as a platform, and also university or research institutes, and potentially some industry partners. So for hospitals, they can provide us enough very high quality samples with very comprehensive disease phenotypes. So, and then BGI will serve as the platform to provide the basic pipelines we streamlined for the for the amplification or enrichment of the repertoire, and also for the data generation. And we also offer the sophisticated analysis pipelines such as a monitor. And for the research institute or universities, they can provide very advanced algorithms or some of the latest pipelines that can do the classification, just as what mentioned yesterday or today, a lot of the machine learning or artificial intelligence techniques that can be combined or integrated. But also some of the universities, we have to provide funding for sustainability of this project. So industry, if they want to translate the discoveries from our cohort, from our data, they have to also provide funding. So in this model, I think that they will provide a sustainable, pipeline for the whole, cohort to be accomplished. So we also welcome other corporations to enter this network. So regarding the current, the Immunome Initiative. So at the end of the 2017, there are almost, actually currently there are more than 20,000 samples inside the Immunome Initiative. So including including the healthy controls, healthy individuals. So for healthy individuals, we have the ages and also gender information, the different ages from the newborn to the elder person. And also we extract collect these samples from different time slots. So they're longitudinal samples. And majority part of this cohort is currently autoimmune disease. So there are almost more than 10 different autoimmune disorders connected. So for each of them, there are several hundred, almost 1,000. And we'll also have some samples in tumor with cancer, and some of samples with different, known allergens, allergy samples, and also some of the infection sample, although the number is not quite large. So because currently we are emphasized a lot on the autoimmune disease, because we think that it may be the most immune-related ones, but we also know that's very difficult because the autoantigens and the autoantibodies are not well defined. So we still want to look at whether autoimmune disease can be a good example to use TCR, BCR repertoire as biomarkers. So currently, we mostly generate data from the T cell receptors, but our next step may be the B cell receptors. So from T cell receptors, firstly, we also did some of the etiology or pathology analysis to looking at the rheumatoid arthritis. So this is a study that we are generated from very small scales of samples, the 12 eye patients and 8 matched controls. So for each patient or controls, we we sorted the different cell types for cytometry, and we sorted naive memory and Tregs and effector memory, and also Th17. In total, there are 8 different type of CD4 T cells extracted from the patient or control samples, peripheral blood. So first, we analyzed the number of the cells in the patients compared with the controls. Of course, as expected, we observed the reduction of the Treg cells and also the increment of the effector memory of Th17 cells. And then we compare, sorry, it's not very, we compare the diversity of different cell types in a patient with the control samples. And we find that the overall diversity measured by different metrics, that in RA patients is lower than the healthy controls in Th17 and also in the effector memory T cells. So then we are try to see whether there's overlap, because Th17 is a very important cell modulator in the eye. So we see whether there's overlap or connection between Th17 cells and the effector memory cells. So we plotted the overlap matrix between the Th17 cells, and all the other cell types. And we found that the overlap in the eye patients between the Th17 effector memory is significantly higher than the control samples. And also this higher correlation between the Th17 and the effector cells. But when we plot the correlation between effector memory cells and other cell types, we only find the overlap, the enhancement of the overlap between Th17 cells and effector memory cells. So this gives us a hint of whether there's a connection between Th17 cells and effector memory cells. So then we did, ex vivo experiment, and indeed we found that in eye patient, effector memory cells are inclined to differentiate into Th17 cells. So from ex vivo experiments, we found that the blood from the patient, the eye patient, that we culture the cells for some time, and the more effector memory cells differentiated compared with controls. And this is not the case in the central memory cells. So this, validates our, analysis. So this is a very simple story about this eye analysis. I don't mention anything else, but this paper is already, in AIRRC4. It's currently under review in some journals. So this is something about the etiology of pathogenesis of eye. But our most interest is not this, as I mentioned to you. So we want to look at, firstly, what's the overall diversity in autoimmune disease? This is open question. And also the gene usage, bias, the preference. But mostly, can we identify autoimmune disease associated T cell receptors and use these T cell receptors for diagnosis in the blood, this non-invasive diagnosis? And what's the features of these T cells? And also, we want to look at the molecular heterogeneity regarding the T cell repertoire in different autoimmune disease, whether we can use it to do molecular subtyping of these diseases. And then what's the implication of this T cell repertoire as biomarker? So in order to do this, firstly, we did a smaller scale project focused on a kind of the autoimmune disease, which is called IgA nephropathy. So in this project, we did, the sample size is not very large, less than 100, but we sequenced both the T cell receptors and both the B cell receptors. And for B cell receptors, we have the IgA nephropathy and also non-IgA nephropathy. So we did a comparison on IgA nephropathy. We found that the diversity of the tissue is significantly lower. But when we compare the peripheral blood, we didn't observe very significant difference between the patient and the controls. And also regarding the B cell, regarding the B cells, the situation is similar, that we observe the lower diversity of the tissues, but when the blood, we even see a higher diversity in both the IgA nephropathy and the non-IgA nephropathy compared with controls. So there's no significant diversity difference. And then we look at the CDR3 NANS. So we look at the out-of-frame CDR NANS of the IgA nephropathy. So we found that the NANS of the overall NANS is significantly shorter in the IgA nephropathy compared with the controls. So this is the implication that whether these CDR3 have any pathology specificity. So most important part, we try to identify some of the disease-related T cells. So in this project, we did very simple. We just look at the public clones. So we look at those public TCRs exist, or TCRs or BCRs existed in a patient, but not existed in the control samples. And we identify some of the TCR and the BCRs, And when we look at their frequencies, we found that in peripheral blood, there are some clonal expansion of these TCRs compared with controls. And also, when looking at the BCR, we have both the IgA nephropathy and non-IgA nephropathy. We found the BCR sequences in IgA significantly higher than the non-IgA and then the control samples. So we then use this public TCR BCRs to do the classification or diagnosis. From small scale samples, we found that for TCR, we have a very good result. This is a leave one out cross validation. So in both the TCR, we identified a very high OC value. And for BCR, we can differentiate IgA nephropathy from the non-IgA nephropathy and also from the healthy controls from their peripheral blood. Although the performance is not that high, but it gives us some, some of the hint or some of the implication. So we truly want to expand our study into larger scale autoimmune disease. And we also want to look at it in a common autoimmune disease, such as the RA or system lupus. So then we did a larger scale project. So this project, including more than 800 SLE samples, all of them generated T-cell receptor repertoire, and more than 200 RA samples, and more than 500, almost 500 healthy controls. So from their population level and T-cell receptor repertoire, we did first a rare fraction analysis, show that even with more than 800 individuals, SLE individuals, we cannot see the overall, this is the unique clone number. So this is a public clone number. For this is public. This is unique clone number. This is public. So from unique clone number, even with the more than 800, we didn't observe a very obvious saturation from the population level. And this is the case for SLE, and this is the case for I and the controls. This is I, this is controls. And when we look at the public clones, the situation is similar, and even less saturation. And then we compare the overlap between the autoimmune disease and the healthy controls. So we can see there are significant overlap between them. And when we see the public clones, we see the public clones overlaps between the disease, the two different autoimmune disease, and also healthy controls is significantly higher. It's higher than the than the other clones, the total unique clones. So this gives us some implication that there may be selection advantage of the public clones from autoimmune disease. So then we compare, well, of course, the very simple overall diversity indices. So we found from different indices that this time we found that the overall diversity in different indices that's in autoimmune diseases is significantly lower than healthy controls, especially in SLE is the lowest. So this is a unique clone number, this is a channel diversity, and this is a expansion of the top clones and some Gini indices. So all the these indices indicate that the overall diversity of autoimmune disease is lower than the healthy controls. So we then look at the public clones. So when we look at the public clones, so this is the X plot is the publicity, is the individuals that harbor the public clones. So we can see that with the increment of the publicity or incidence of individuals, these clones, the frequency of these clones in the population is become higher. So this is the case in RA. We can see the I is significantly higher. So in this plot, we can see I is significantly higher than the healthy control and SLE. And SLE is higher, but it's not that obvious. So we can see the public clones in autoimmune disease have a clonal expansion. And the higher of the publicity, the higher the clonal expansion. But this clonal expansion is not that obvious maybe compared with the other infection disease. But of course, there's a clonal expansion. So we then try to use a model, try to identify this disease associate or autoimmune disease associate TCRs. So we're firstly doing a statistical test to every T cells in the autoimmune disease to see their incidence observed in individuals and also their frequency. So we integrate both the publicity of these T cells and also their frequencies. So our hypothesis is that if the publicity of this clone is higher in the autoimmune disease, then the controls is a higher probability to be the autoimmune disease associated clones. So in this way, we identified a batch of TCR receptor sequences. We determined them to be autoimmune disease associated or specific. Then we also have a weighing value for these TCR sequences. Then we have a random forest model to do the classification using these disease-associated TCRs. So firstly, that we evaluate whether these TCL clones truly have some association with the disease. So we don't have we didn't do any experimental validation on this, but we try to figure out whether there's a connection between the phenotypes. So we look at the disease-associated clones, the content in correlation with the disease activity. So we found a very linear, correlation between this disease associated clones with the activity of the disease, and also measured by the complement 3 in SLE. And for I, we also identify the correlation between the disease-specific clones associated with the disease activity measured by DS28 and the ESR. So this gives us a hint that these clones should be correlated with the disease. But then we also look at the using the TBI-DB, just I mentioned that the manual curated database and some other like the VTJ-DB databases. So we look at those databases and look at the annotated SLE or I associate clones, their appearance in our data set. So we identify some of the clones, not too much in our databases, but none of these clones existed any increment of the publicity. So none of them have been identified in our disease-associated clones. So things that this phenomenon has also been observed in other studies, such as the CMV study. So there's some explanation for this, although the result is disappointed, but some explanation existed, such as the currently in the database, annotated database, the clones is not that comprehensive. And also we can see the TCR privacy can contribute a lot to this phenomenon, and also some HLA bias. So in summary, we didn't observe any database annotated, previously reported autoimmune disease related TCRs observed in our dataset. So then we use our in silico identified TCRs to do the diagnosis or classification of the autoimmune disease. So we found that if we look at the SLE and the controls, so we can observe a very good performance of classification between SLE and the controls. So OC value can be more than 94%. So the frequency is pretty high, including sensitivity and specificity. And regarding the RA and the controls, also the OC value is pretty high. Or this is RA and the control performance. And we can also efficiently classify SLE from I using our identified disease-associated clones. So then we look at the properties of our in silico identified disease-associated clones. So of course, we identify the clone frequency of the disease-associated clones is a little bit higher. That's as expected in both SLE and I. And we also look at the CDR3 length. We found a lower CDR3 length in SLE, but not in RA. And we then identified the motifs. We found that SLE and I share much more motifs than other disease-unrelated clones. Sorry? Okay. So yeah, very quick. So then we look at using Levinson distance to cluster all these clones. So we found that this is including both SLE and I associated clones we identified. We found a lot of the associated clones can be clustered together. And the red dots including the clones that are shared by both, both SLE and the I associated clones. So they are placed in the major clusters. And when they do the disease unrelated clones, we didn't observe this phenomenon. We also use the GRIEF software package to do analysis. We found the SLE associated clones can be clustered together, but I associated clones are not that obvious. And we compare the similarity of the disease associated clones with the naive cells. This is the paper published in Nature. So we found a closer similarity, higher similarity in disease associated clones compared with the naive in both SLE and I. So a very interesting phenomenon we want to see is that is there any universal disease associated clones in the entire autoimmune spectrum? So we compare the disease associated clones in I and SLE. So for all the disease associated clones we identify, there are 32 can be observed in the I, and 12 of them is both I and SLE associated clones. So we also identified other clones, the 32 clones we found, although they are not identified as SLE associated, but they occur with higher publicity, with high incidence in SLE than the controls. So means they have a higher probability to be SLE associated. So these results suggest that there should be existence of the autoimmune spectrum, existed associated clones. So there may be similarity of the autoantigens or maybe similarity of the autoantibodies in the common autoimmune disease. Okay, then we look at the association of the properties, both the disease-specific clones and overall diversities with the clinical incidences. We found a lot of correlation in the overall diversity and overall incidences, but the most significant phenomenon result we identify is the disease-associated clones, that most of them have been associated with the clinical phenotypes. Okay, so lastly, we try to use the TBA-DB to find out the annotated sequences in our database. And we found out our T-cell repertoire contains a lot of the pathogen-related clones and autoimmune-related clones. So this is the enrichment of pathogen-related clones in our dataset implies there may be some of triggering from the pathogen-derived clones and some of the sharing of epitope between both the pathogen-related autoantigens and also the autoimmune disease. And sharing with the other autoimmune diseases is expected. So this paper has been recently accepted and is under production in a clinical journal, which is Annals of Rheumatic Disease. So this is I don't know whether you know this journal, but this one is the top ranked journal in rheumatic disease. So I think publish such kind of paper in a clinical journal means a lot to us. So I think at least it can educate the clinicians that this has the potential to be the clinical biomarkers. So we put the title very simple and straightforward. The T-cell receptor repertoires as normal biomarkers. No complex, very simple. So we want the clinicians try to translate it and provide more samples to validate results. So okay, so this is our group I want to acknowledge. So my group, including our collaborators. So that's all for my talk. Thank you. Any questions?
Hi, do you have any studies underway or planned on the 20,000 cohort that you can talk about?
Sorry.
You say there are 20,000 samples in the PRD.
Yeah.
Or do you have any interesting studies that you're working on that you can talk about?
Yeah. That's the ongoing project. I think Science already mentioned this in morning. So these are some of the early results we generated as we are, but for the total cohort, because we are finished the sample generation just recently, so we started still working on it, And also, there's one thing that our collaborators, because they're from different hospitals, they provide samples, so they want to have the priority to doing research. But eventually, all the data, will be shared and after publication. So that's the case. Yeah.
All right, let's thank all our speakers.